Okay, you're looking at a chart that sort of shows you how it is hormones carry out their task, and you can see at the top of the flow chart, hormones can act on their target tissue by binding to either membrane or nuclear receptors. The hormones that bind to nuclear receptors are going to be those that are lipid soluble. So let's stipulate that on the chart. So these are lipid soluble or hydrophobic hormones. So those would be the catecholamines or the steroids, and over here, water soluble. What happens next is that the ligand attaches to its receptor. In the case of water-soluble hormones, will alter the activity of a second messenger of some type. Now, in many cases, this will be a G protein. Sometimes these water-soluble hormones will bind to receptors that change the activity of an enzyme on the cytoplasmic side of the cell membrane. Next, we'll either open or close ion channels and activate enzymes inside the cell in the case of a G protein linked receptor, or in the case of a receptor that activates intracellular enzymes, we'll activate those enzymes and generate a cell response. For the lipid-soluble hormones, we're going to activate certain clusters of genes as a result of the hormone receptor complex binding to the promoter and, along with other transcription factors, turning the gene on or off and producing or shutting down the production of a particular gene product, and that's what will generate now the desired response in the target tissue. A portion of the molecule where the hormone binds is called the binding site. The receptors are proteins that are going to have a cleft on the surface that's going to fit perfectly with the ligand. If the molecule is a receptor, like in a cell membrane, the binding site is called the receptor site. If the hormone receptor site is specific, such as in the case for epinephrine, it cannot bind to the receptor site for insulin. So we have to understand is what defines a target tissue is the presence or absence of a receptor specific for that particular hormone. Now, this brings up an important point, which is that the target tissues can adjust their sensitivity for a particular chemical signal by changing the number of receptors either on the membrane surface in the case of water-soluble hormones or inside the cytoplasm of the target tissue in the case of the catecholamines or the steroids. And this happens as a result of increasing or decreasing amounts of the hormone as conditions in the body shift. Generally, when less hormone is secreted, receptor concentration goes up so that the sensitivity of the target to the ligand increases, and when amounts of the circulating hormone increase over time, the receptor concentration to that ligand will decrease. And this is a way to modulate, again, the response of the target tissue to the ligands so that you do two things at once. Number one, you maintain response to the chemical signal, while at the same time you don't cause so much of the cell's machinery to be involved with responding to the hormone that the cell itself, the target tissue, can't survive. If you overload the system, what can happen over time is the target tissue can completely lose its sensitivity to the hormone, and the result will be that body's homeostasis will be disturbed. And an example of that is type 2 diabetes, in which as a result of repeated production of insulin is an outcome of a lifestyle in which you constantly consume calories without burning them, over time target tissues downregulate their concentration of insulin receptor, and we eventually become insensitive to our own insulin. The fix for this is to consume fewer calories and exercise more and give the target tissues a chance to recover over a fairly significant time period. Eventually what happens is normal function is restored in most cases and everything goes back to normal. The purpose of binding to the target tissue is to elicit the response by the target cell. This creates conditions in the body that eventually turn off the original chemical command, the original endocrine secretion from that organ. So this is just telling you a little bit about how the cell can modulate receptor concentration. Normally, receptor molecules are broken down and replaced on a regular basis. In downregulation, what happens is that the rate at which they're broken down is greater than the rate that they're synthesized. A combination of hormones and receptors can increase the rate at which the receptor molecule is broken down, and this combined form is taken into the cell by phagocytosis, broken down, and then the component parts are recycled. In upregulation, some stimulus causes an increase in the synthesis of receptor for a particular hormone and increases the sensitivity of the cell to that chemical signal. An example is follicle-stimulating hormone. It stimulates the ovary to cause an increase of luteinizing hormone receptors. The ovarian cells are now more sensitive to the luteinizing hormone even if the concentration of LH doesn't change, and this is what triggers ovulation. So this is an example of a synergistic effect of two hormones in order to improve the response of the target tissue. So one sets the table for the other. Remember that there are different classes of receptors, that the lipid-soluble hormones are going to tend to have receptors that are either in the cytoplasm or the nucleus of the target cell, while water-soluble hormones are going to have receptors that are on the membrane surface. So you can see here that with a lipid-soluble signal, we can very easily pass through the cell membrane and bind to the receptor either in the cytoplasm or in the nucleus itself, and then as a result of that complex being formed, we search out the target gene and we trigger transcription and as a result translation. Examples of hormones that work through this method are thyroid hormone, testosterone, estrogen, progesterone, estrogen, aldosterone, and cortisol, all lipid-soluble hormones. You can tell by looking at their structure, because they're predominated by carbon and hydrogen, that they're not going to dissolve very well in water. Remember, the one way to determine whether or not a molecule is going to be water soluble is to look at its chemical structure and scan it for the presence of electronegative atoms. And when we're talking about biological molecules, electronegative atoms are going to be things like nitrogen, sulfur, oxygen, and phosphorus. Basically, elements that are to the right of the carbon and to the left of the noble gases in the periodic table. Remember that water-soluble hormones have receptors on the cell membrane. What happens when they interact with their appropriate membrane, the hormone is going to trigger either their activation of a second messenger or of an enzyme downstream. That'll result in a cascading effect that'll eventually produce the response desired. Remember that water-soluble or large molecular weight polar hormones cannot pass through the cell membrane. As a result, they attach to their receptor on the membrane surface, and this results then in a cascade of events that leads to response of the target tissue. Examples of hormones that have receptors on the cell membrane surface are large proteins, glycoproteins, and polypeptides, and smaller molecules like epinephrine and norepinephrine. So you can see the model here, which invokes a G protein. Right? Ligand-receptor interaction results in a conformational change. The conformational change activates the associated G protein. Subunits of the G protein activate adenylate cyclase. Adenylate cyclase causes the formation of cyclic AMP from ATP, and this now has a downstream effect. Could be on a protein kinase, could be on other molecules downstream in the cascade. The point of the cascade, remember, is that at each step in the cascade, the activated component can activate more than one downstream target. The result is that the signal amplifies at each step, and then very quickly a small stimulus is amplified to a very large stimulus, which produces the cellular response. The other important aspect of a cascade is that it's easy to turn off because all you have to do is inactivate the first or second event in the cascade. And this is just a look at some different hormone categories, their receptor types, some examples, and their mechanism of action. That's 17.3 in your text. Just make sure and review that material. So let's talk about these nuclear receptors first. These are proteins that reside in the cytoplasm or the nucleus awaiting the arrival of the lipid-soluble hormone. The hormones bind with the receptor on the cytoplasmic side of the cell membrane. This complex then in the nucleus activates particular genes, triggers transcription, the message is then translated, and the production of that protein product is basically the response of the target tissue. A latent period of several hours occurs because there's time required to produce the message in the protein, and the process is limited by breakdown of receptor hormone complex. Estrogen and testosterone produce different proteins in cells that will have receptors for either of these hormones, and this causes the differing secondary sex characteristics in males and females. So remember that a target tissue is defined by the presence or absence of the appropriate receptor on the target or in the target, and a single cell or tissue can be a target for more than one type of hormone, and this is the example here. So let's take a look at how steroid hormones work.
Steroid hormones are not water-soluble. They travel in the blood attached to protein carriers. When steroid hormones arrive at their target cells, they dissociate from their protein carriers and pass through the plasma membrane of the cell. Some steroid hormones bind to specific receptor proteins in the cytoplasm and then move as a hormone receptor complex into the nucleus. Other steroids travel directly into the nucleus before encountering their receptor proteins, not shown. The hormone receptor protein, activated by binding to the hormone, is now able to bind to specific regions of the DNA. These DNA regions are known as the hormone response elements. The binding of the hormone receptor complex has a direct effect on the level of transcription at that site. Messenger RNA, mRNA, is produced, which then codes for the synthesis of specific proteins.
Okay, membrane-bound receptors are essentially on the phospholipid bilayer surface. The binding site is going to be on the extracellular side, and the portion of the membrane-bound receptor that is going to trigger the cellular response is on the cytoplasmic side of the cell membrane. These are intracellular mediators. Generally, ions or molecules enter the cell or are produced in the cell as a response to the binding of the hormone. This can be produced because of activation of a G protein. We can also regulate intracellular enzyme activity through something such as a kinase cascade, where a series of phosphorylations triggers the transformation of a cytoplasmic protein from an inactive to an active form. Okay, this is just a look at some common intracellular mediators. The mediator is shown in the left, the example of the cell type in the middle, and the response here on the right. Just star this and know it. This is 17.4 in your lab manual. Okay, let's take a look at how it is that membrane-bound receptors activate G proteins.
The membrane-bound receptor has a receptor site for a small molecule, called a ligand, exposed on the outside of the cell. The portion of the receptor on the inside of the cell can bind to the G protein, which has a guanosine diphosphate attached to its alpha subunit. When the ligand binds to the receptor site on the outside of the cell membrane, the G protein changes conformation, and guanosine triphosphate replaces the guanosine diphosphate on the alpha subunit of the G protein. The activated alpha subunit then separates from the beta and gamma subunits. This step can be repeated as long as the ligand remains bound to the receptor. When the ligand separates from the receptor site, additional G proteins are no longer activated. Inactivation of the alpha subunit occurs when its own phosphorylase activity removes a phosphate from the guanosine triphosphate, leaving guanosine diphosphate bound to the subunit. The G protein subunits then recombine and attach to the receptor in the cell membrane.
And let's take a look now at how membrane-bound receptors, G proteins, and calcium channels interact.
When a ligand binds to the receptor site on the outside of the cell membrane, the G protein changes conformation, and guanosine triphosphate replaces the guanosine diphosphate on the alpha subunit of the G protein. The activated alpha subunit then separates from the beta and gamma subunits. The alpha subunit, with guanosine triphosphate attached, binds to the calcium ion channel, causing the calcium ion channel to open. Calcium ions diffuse into the cell and combine with calmodulin. The combination of calmodulin and calcium produces the response of the cell to the ligand. Phosphorylase activity removes a phosphate from the guanosine triphosphate, leaving guanosine diphosphate bound to the alpha subunit. The inactivated alpha subunit separates from the calcium ion channel, and the channel closes. The alpha subunit recombines with the gamma and beta subunits, and the G protein recombines with the receptor.
This diagram is just showing you an example of a G protein second messenger system in which the hormone binds to its receptor on the surface and activates a cascade of events downstream. So here we can see the example given as epinephrine, and what happens when epinephrine binds its plasma membrane receptor is that it will trigger now the activation of a G protein which is associated with that receptor. The G protein basically decouples from the receptor site, and the alpha subunit is now going to trigger, in this case, the production of a second messenger in the form of phosphoinositol bisphosphate. That ends up triggering now eventually the activation of cytoplasmic enzymes through a kinase cascade, and also triggers the release of calcium from inside the cell via alteration in the conformation of calmodulin. Okay, so the inositol phosphate releases calcium from the ER. Remember, the smooth ER is a calcium storehouse, and also diacylglycerol can regulate enzymes such as prostaglandins and other protein kinases, and this will cause an increase in the strength and the duration of muscle contraction. So these two act together. These are two different branches of the exact same cascade. Okay, but the bottom line again is that at each step, each participant in the cascade can activate more than one downstream target. Okay, so again, just to follow, right? Hormone receptor binding, G protein activation, phospholipase C triggering production of PIP. PIP is released, inositol phosphate becomes one branch of the second messenger pathway, and the diacylglycerol becomes the second branch. The diacylglycerol activates protein kinases that leads to the activation of intracellular enzymes. The inositol triphosphate causes the release of calcium from the ER. The calcium can bind to calmodulin, and that helps to enhance the response. Let's take a look at how cyclic AMP acts as a second messenger in these receptor systems where hormone binds ligand, and we have a conformational change that results in the activation of adenylate cyclase. This is again a G protein modulated system.
When a signal molecule, such as epinephrine, binds to a cell surface receptor protein, it activates a G protein on the inside of the cell. The G protein then stimulates adenylyl cyclase to produce large amounts of cyclic AMP from ATP within the cell. The cyclic AMP then binds to and activates a target protein, such as alpha kinase, which adds phosphates to specific proteins in the cell. The effect of this phosphorylation depends on the identity of the cell and the proteins that are phosphorylated.
So just to revisit, notice again that we have to have binding of the hormone to the receptor, causing the receptor to change its shape. This causes the production of cyclic GMP from GTP via the enzyme guanylate cyclase. The cyclic GMP is then going to act as an intracellular mediator. It'll alter the activity of downstream target enzymes, and this produces the response in the cell. Phosphodiesterase is the enzyme that acts as the shutoff for this cascade once we stop the initial event, which is the binding of the receptor to the hormone. So we have to have a way of turning off the downstream targets, and the phosphodiesterase will cut off or reduce the concentration of second messenger, and as a result the cell response will abrogate. There are also receptors that are going to phosphorylate intracellular proteins directly. In this case, the hormone is going to bind to its membrane receptor. Part of the receptor protein on the inside of the membrane is an enzyme that's going to phosphorylate a downstream target. The phosphorylated enzyme is now considered active and will lead now perhaps to the phosphorylation of other downstream targets, which will activate other downstream targets, and eventually what will happen is that you'll amplify the response to the point where you'll generate the desired reaction of the target tissue. An example is insulin. They bind their receptors, causes phosphorylation of proteins, and the cell then responds appropriately to the presence of insulin. Generally, this means that the cell is going to synthesize membrane proteins that are going to transport glucose into the cell for use as energy until such point as the insulin signal is no longer produced. At that point, what will happen is that the change in conformation of the membrane receptor will cause the phosphorylation of the downstream target to cease, and at the same time there are going to be phosphorylases that are going to cleave the phosphate groups from the activated downstream enzymes, and the entire response will stop. And this is just showing you the principle that we've been discussing when we talk about cascades. In class, I asked you to remember a game you may have played when younger, which was freeze tag, where if you were it and whoever you touched, they ended up not being able to move. The result was an amplification of your effect on the rest of the people playing the game. Same thing really in a cascade inside the cell. We have an activated first event leading to more than one downstream participant in the cascade being activated, which can activate more than one downstream participant at the next point in the cascade. And you can see how the number of participants in the cascade event doubles, triples, amplifies at every step until we have a giant pool of activated downstream targets that produce the effect desired in the tissue. Okay, this is just a look at signal amplification and the principle behind it. Keep in mind that cascades aren't just something that you're going to see inside cells and target tissues. You're also going to see it extracellularly. Blood clotting is an example of an extracellular cascade, as is inflammation. So take a look at this and then remember, review this material as you will see it on the next PRS quiz. Thank you, everybody.
Receptor proteins interact with signal molecules at the surface of the cell. In most cases, the signals are relayed to the cytoplasm or the nucleus by second messengers, which influence the activity of one or more enzymes or genes inside the cell. However, most signaling molecules are found in such low concentrations that their effects in the cytoplasm would be minimal unless the signal were amplified. Therefore, most enzyme-linked and G-protein-linked receptors use a chain of other protein messengers to amplify the signal as it is being relayed. In the case of a protein kinase cascade, one cell surface receptor activates many G-protein molecules. Each G-protein activates many adenylyl cyclases. Each cyclic AMP in turn will activate protein kinases, which then activate several molecules of a specific enzyme. The end result is rapid production of high levels of the final product.